Right, hi there. I'm going to work through some exam questions, some examples, and then you can use this information and how I would do it to have a go at answering some similar ones that are on class charts. So the first one is about testing leaves. Okay, now normally when we do the test, it's a test for starch. Okay, and we'd use iodine. Now in this question, they talk about a variegated leaf, which is a two-coloured leaf. It's left in the sunlight for several hours, so it's going to be photosynthesising. That's the idea that they're getting out with the sunlight. And then they've taken the leaf off and tested it for sugar. Okay, now the point with this is that if it's producing sugar, then it's doing photosynthesis. Okay, so that's what we need to take from our results, and it gives you the information, and these are the results here. Okay, so you've got the paler part around the outside and then the green part on the inside. Okay, so this is a clue. Now, it then tells us that this part from the paler area in the leaf doesn't contain any sugar. Okay, and the piece that's been taken from the green area does contain sugar. Now the question says, explain as fully as you can why the yellow region had not produced sugar. Okay, so the point with this is that it's obviously lacking something that is making this part of the leaf green. Okay, and that something is chlorophyll. Okay, so the yellow part has no chlorophyll. Has no. Okay, so that's explaining why it's not green. And then we need to say, well, why is that not allowed it to photosynthesize and produce sugar? Because if it's sugar, it's getting at the fact that the plant is or isn't doing photosynthesis. So the yellow part has got no chlorophyll, which absorbs light for photosynthesis. Okay, so if it can't absorb light, it can't do photosynthesis, it can't produce sugar. Okay, so that's what, sometimes you get variegated leaves, sometimes you get a leaf where there's been a section blanked off. It's all looking at exactly the same idea. If it's not got sugar or starch in it, there's something that's preventing it from doing photosynthesis, and you just have to identify what that thing is and then say that it hasn't got this, so it can't do photosynthesis. Okay, so this exam question looks at an experiment investigating the rate of photosynthesis, so the speed of photosynthesis, how quickly it's happening. Okay, now in this experiment, and it comes up in lots of different ways, we always use pondweed. Okay, now we could test the leaf for starch and that proves that photosynthesis is happening, but we can't use that to tell us how quickly it's happening. So what we use instead is the oxygen that's given off. Okay, that's why we use a pondweed, because as the oxygen is given off, it forms bubbles in the water, and we can either count the bubbles or we can actually measure the volume of gas that is collected at the end of the tube. Okay, so it's to do with speed in terms of photosynthesis happening. Now this question asks us, sorry, I'm just having camera difficulties. This question asks us about different colours of light. So it's asking us to investigate how it's affected by different coloured lights. So that is the thing that I am going to change. Okay, now what we need to do is describe the method that we'd use to what we would measure, okay, and what we would control. So if you're controlling it, these are the things that you need to keep the same. Okay, so any experiment is always following exactly the same formula. It's chemistry or biology. You need to think about what it is you're changing, what it is you need to keep the same, and what you will measure for your result. Okay, now it's a 6-mark question, so I need to make sure that I've included a number of different points. Okay, but having those 3 actually helps me to structure how I'm going to approach my answer. So I've got my diagram here. It's sort of half given me what I'm going to do. Okay, so my first point is set up the equipment as shown. Now if I'm testing different colours of light, I must somehow have a lamp near to my pondweed. So I'm going to set up the equipment as shown with a lamp Now this is one of the things that I need to control, and I need to keep the distance the same, okay, with the lamp to the beaker. So I'm gonna give a distance. So I'm going to say the lamp is at 20 centimetres from the beaker. So I've already listed something that I need to control, okay. Then what I'm going to do is I'm going to measure. So what am I measuring? I can either measure the volume or I can, sorry, let me just turn my phone off. I can either measure the volume or I can count the bubbles. Now counting bubbles is probably the easiest thing, okay, but I need to be specific in terms of how long I'm counting them for. So again, I'm just going to pick a time, right? So I'm going to count the number of bubbles in 2 minutes. Now you could say 1 minute, you could say 5 minutes. It's entirely up to you. Okay, but I've said something. So this is something I'm keeping the same. This is something I'm keeping the same. And then I'm measuring my number of bubbles. So I'm doing it for 2 minutes. Okay. Now, so that I can have valid results that are reliable, that I can trust them, particularly in biology, we need to repeat it. Okay. So I'm going to repeat this experiment 3 times and calculate the average number, oh I missed my A out of average there, number of bubbles. Okay, so I've got a much more reliable result if I can work out an average. Now I need to go back to the thing that I'm going to change. Okay, so I've kept some things the same, I've measured something, but I haven't changed anything yet. So I need to change the different colour of light. Okay, so what my last point would be is that I'm going to repeat the experiment with different coloured bulbs or filters that you can put in front of the lamp so that you can get the different coloured light on your pondweed. Okay, so now I've written 4 points, but I've included all the parts that they've asked me to do. So I've said what I'm going to change, I've said what I'm going to measure, that's the number of bubbles, and I've said some things that I'm going to keep the same. Okay, now this experiment can also come up as investigating light intensity, and what you would do is exactly the same, but instead of changing the colour of the light, what you would do is that you would have your lamp, sorry this is a really badly drawn lamp, you'd have your lamp next to it, and what you would do is you'd move it further away. Okay, so that's an alternative way of doing this experiment, but you are very likely to see something like this, okay, and be asked to write a method, right, but as long as you follow exactly the same formula. So what is it you're changing, what is it you're measuring, and what is it you're keeping the same, okay, then you will get a good number of marks. Okay, so that's how we write methods for experiments. Okay then, so this last question that I'm going to work through for you is thinking about plants and their sort of interaction in terms of photosynthesis and respiration. Now you need to remember that plants don't only do photosynthesis, okay, they also respire, and respiration is sort of the opposite of photosynthesis. Okay, so where photosynthesis uses carbon dioxide and makes oxygen, respiration is the opposite to that. It uses oxygen and it produces carbon dioxide. So any question when it's talking about, it's saying in a greenhouse, it's talking about plants, right, and it's looking at carbon dioxide. So you need to think about straight away, is it talking about photosynthesis or is it looking at the process of respiration? Okay, because those two always kind of go together. So we've got a period of 24 hours. So the first thing I do, I'm going to circle my 2 axes so that I know what it is that I'm looking at. So I've got my time across the bottom and I've got my concentration of carbon dioxide up the side. So before I've even looked at the question, I've thought about what the information is showing me. Okay, now again, at the graph, what we need to think about is the specific times of day that are labelled. Okay, so thinking about these two processes, we need to think about what is happening at these times of day. So it's midnight, okay? It's going to be dark, right, at both ends, because that is relevant to these two processes. At midday, there is light, okay? So these, just by, like I said, just by thinking about the graph first, it's going to help me in the questions itself. So it says, explain why the concentration of carbon dioxide in the air in the greenhouse increased between X and Y. So we're looking at carbon dioxide, okay, and we're looking at between X and Y. So if I look at X, it's midnight, it's dark, right? So it's not going to be photosynthesis. Okay, so straight away I can forget about photosynthesis. Something is happening in terms of respiration, right? So my plant is respiring and not doing photosynthesis, okay? So as it goes through the night, what I can put for this is the plant is respiring and respiration. So sorry, I'm talking and writing in different words. I'm producing carbon dioxide. Okay, the plant is doing respiration. I've identified that, and I've identified the link between the respiration and the carbon dioxide, why that's going up. Okay, now then it asks me, explain why the concentration of carbon dioxide in the air decreased between Y and Z. Okay, so I go back up here. I've got Y, dawn is the beginning of the day, okay? And then it's going into sort of late afternoon. So if we're in the period of time when it's light, I need to be thinking now, out of my two processes, it's going to be the photosynthesis, okay? So what I need to think about now is, well, why is carbon dioxide going down if the plant is doing photosynthesis? Carbon dioxide is one of the reactants, one of the ingredients that you need for photosynthesis. So what I need to think about in that case is that the carbon dioxide is being used by the plant for photosynthesis. Okay, now what we need to remember is that the plant is also still respiring. It's not that it only does photosynthesis and only does respiration at night. Okay, it's still respiring. So the last point for this is that there is more photosynthesis, because it will be producing a little bit of carbon dioxide. So there's more photosynthesis than respiration. Oops, got spell. Okay, so that's why the carbon dioxide is going down, because there's more photosynthesis than respiration. It's producing a little bit, but it's using more than it's producing. Okay, I'm just going to try and stop my video.